Hi, this is Dr. Claire, and this is a lecture on characteristics that we use to classify animals into groups. So we'll be going through a number of different characteristics that you can use to figure out what kind of an animal you're looking at, basically. All right. So to start with, what we usually look at are things that are fairly conserved, things that don't change very rapidly. So the two major types of information that we use to classify animals are usually developmental patterns and body plans. So we'll be going through each of these individually. So we'll start with those developmental patterns. So one really important way that you can classify animals is by looking at the number of different types of embryonic tissue layers they have. So some animals have only two types of embryonic tissues, ectoderm and endoderm. Ectoderm means outside skin, endoderm means inside skin or inside tissue. And so a diploblastic creature has just those two, ectoderm and endoderm. So there's one that's on the outside, it's an embryonic tissue, it forms the skin, and one on the inside, it's an embryonic tissue that forms the gut, okay? So that's diploblastic. An example of a diploblastic animal are the cnidarians, jellyfish and sea anemones. Most other animals are actually triploblastic, meaning that they have three embryonic cell layers. They have the ectoderm, they have the endoderm, and then they also have one in the middle, not surprisingly called the mesoderm, so middle skin or middle tissue, right? And in the case of a triploblastic creature, the ectoderm generally forms the skin and the nervous system, the endoderm forms the digestive system, and the mesoderm forms pretty much everything else, so your bones, your muscles, and whatnot, okay? So those are the two major types. You should note that sponges are weird, which I will be repeating a lot in this lecture. Sponges have no distinct cell layer types. So when we talk about sponges, you're going to find that they are really quite a different critter. Another thing you can characterize animals by is the first few cell divisions, the patterns in which those cells divide. Those are called cleavages. Yeah, I know, cleavage. It's the name that they came up with. So the first few cell divisions are called a cleavage. So you have your first cell cleavage that divides the single fertilized egg into two cells. The second cleavage divides that into four. And then the cleavage that we're interested here is actually the third cleavage, which goes from a 4-cell embryo to an 8-cell embryo. So each of those cells in the 4-cell embryo divide in half. And there's two types of cleavage that you can have. You can have spiral cleavage or radial cleavage. In spiral cleavage, when the 4 new cells divide off the bottom cells, they're rotated slightly so that they're offset on top. So it's kind of forming a spiral. Radial cleavage, the 4 new cells just stack right on top of the old cells. So mollusks and annelids, for example, have spiral cleavage, whereas chordates, such as vertebrates and echinoderms, have radial cleavage. All right, one more developmental pattern is gastrulation, the pattern of gastrulation. So once the embryo divides for a while, what you get is a ball of cells. So it's basically a hollow ball of cells. That's called the blastula. And then the blastula goes through the process of gastrulation in which the digestive system starts to form. And so what happens at the beginning of gastrulation is that an opening in that ball of cells starts to form. So you get like a dimple on one side. That's called the blastopore. Now, the fate of the blastopore can be different. So remember, this is the first opening to the digestive system. In most animals, the digestive system is a tube. In fact, in all triploblastic animals, the digestive system is a tube. And so the first opening to the digestive system is either going to form the mouth or it's going to form the anus. And so if the first opening, if the blastopore becomes the mouth, then that animal is called a protostome. Proto means first, stome means mouth, so mouth first. And so some examples of protostomes are the mollusks and the arthropods. The other option is that the first opening forms the anus instead of the mouth. And those are called deuterostomes. Deutero means second, stome means mouth, so mouth second. Or you can remember it as deuterostomes. The anus forms first, ha ha ha, poop jokes. And either way, that's the other thing that can happen. So you either have your mouth form first or you have your anus form first. Animals that are deuterostomes include the vertebrates, such as ourselves, the chordates, and the echinoderms. Okay? All right, so the other types of things that we use to classify animals into groups are the body plans that these animals have. So the general structure or the arrangement of the animal. And so we're going to be talking about symmetry, body cavities, so empty spaces in the body, and segmentation. So we'll start with symmetry. Most animals have some sort of symmetry. Symmetry basically means that you can divide the animal in half in some plane, and the two halves of the animal are identical to each other. So the two major types of symmetry that we have are radial symmetry, where you can divide the animal, it's like a pizza, you can divide it in a whole bunch of different planes and you get two equal halves. So like jellyfish and sea anemones have radial symmetry. Most other animals have bilateral symmetry, where there's only one plane that you can divide the animal in half and you get two equal halves. So that would be bilateral symmetry. We're an example of a bilaterally symmetrical animal. You can divide us this way and you get two equal halves. Okay. So what's so great about bilateral symmetry? It's how come so many animals have bilateral symmetry? Well, it turns out having bilateral symmetry gives you the ability to have directionality. So if you are a radially symmetrical thing, you don't really have a forward and a backwards. You're kind of round, right? So you could go in any direction that you wanted. But that doesn't, they kind of just drift around and float. You know, they don't have a real purpose to their movement. Bilateral symmetry, what you get is you get a directionality to the animal. So the animal's going to move in one particular direction, and you tend to get cephalization, so that concentration of nervous system and sensory organs in one end of the animal, the brain, right? And that's usually the front end of the animal, which is the first part of the animal to come in contact with the environment, which is where you want all your sensory organs to occur, right? Okay, are there any asymmetrical animals? Yes, there are. Sponges are weird. Sponges are asymmetrical. So they just kind of form associations between the different cells in the sponge, and they can kind of form whatever shape they want. So they don't have any particular shape. They have various kind of standardish shapes, but none of them are symmetrical. There's no way you can divide a sponge in half and get two equal halves. Okay. Body cavities. So a body cavity is basically a space within the body that's not in the digestive system. It's an area where there's no tissue. Generally, it's a fluid-filled area within the body of the animal. Okay, so it's between the digestive system and the body wall, generally. So not all animals have body cavities. There are some animals that do not have a fluid-filled space within the body. Those are called acoelomates. A means without. Coelomate means a fluid-filled body cavity. Coelom is a fluid-filled body cavity. All right. So acoelomates include things like flatworms. There are also animals that are called pseudocoelomates. Pseudocoelomates do have a fluid-filled body cavity. The phrase pseudo means fake, but that's a little bit misleading because they do have a fluid-filled body cavity. It's just between the endoderm and the mesoderm. So they don't have muscular control over their endoderm tissue because the mesoderm forms the muscle. So they have this gap between the muscular outer body and that digestive system. Okay, so that's a pseudocoelomate. A coelomate, or a true coelomate, has a fluid-filled body cavity that's completely surrounded by mesoderm. So there's a mesoderm layer that surrounds the endoderm, and then it goes out to the body wall, and there's a cavity that's within that mesoderm tissue. That means that there's muscle inside and outside the fluid-filled body cavity, which means that the animal has more control over that fluid because it can contract muscles on both sides of it. Okay? So roundworms, or the nematodes, are an example of a pseudocoelomate. The annelid worms, and also ourselves, are examples of true coelomates. All right, and finally, segmentation. So segmentation is a type of organization of the body where you have repeating segments that have similar structure repeating over and over again. So what this allows for is specialization of different body regions for particular functions. So for example, the arthropods have these repeating segments, and some segments are specialized for reproduction, and some segments are specialized for movement, and some segments are specialized for feeding. So they have this ability to have different functions for those different regions. As vertebrates, we also have some segmentation. Our spine, for example, with the repeating segments of the bones in our spine, allow for segmentation. The other thing about segmentation is that it usually makes you very flexible, so we can bend like this and back and forth. So those are some of the benefits of segmentation. All right, so all of these different traits allow us to classify animals into a phylogenetic tree. And so you can look on the tree and see where these different traits arise. So you have the specialized tissues here. So here's the sponges down here at the bottom. Sponges are weird. And then you have the evolution of specialized tissues. Radial symmetry breaks off here and bilateral symmetry there. And then you have your triploblastic, your third tissue layer. The protostomes and the deuterostomes are another major division. But all animals are related to each other. Animals are a monophyletic group. Cast your minds back to evolution. You hopefully remember that a monophyletic group is a group that contains the common ancestor and all descendants of that ancestor. So we are a monophyletic group. We are more closely related to all other animals than we are to any other group that's alive today. Okay. Check out the next lecture for the diversity of animal groups.